ITI Academy Learning Module, An Introduction to Biostatistics by Tony Dawson. The one constant in the biological population is variability. It's very unlikely that two individuals will be identical in every way. We can see this demonstrated in this image of the participants at the 6th ITI Consensus Conference in Amsterdam. This group of individuals varies considerably across a wide range of criteria. This variability between individuals is the basis of natural selection and evolution. Variability can influence many biological processes, including how patients will respond to disease and how they might react to the treatments that we provide to them. If we wish to determine if our treatments are effective, we need to consider how variation in the population might affect our results, and whether the outcomes we detect are due to our interventions or just the effects of blind chance. Biostatistics is the body of knowledge that we can use to measure variability and describe the average individual, that is, to describe the central tendency of the population. By statistically comparing these features of two or more groups of individuals, we can help determine the root cause of the effects that we observe. You may wonder why biostatistics is important to us as clinicians in our daily practice, but it does matter. Our treatment usually works, but sometimes it does not. Dental interventions are not as predictable as machine repairs because of human variation. One successful case does not necessarily guarantee that the procedure or material will work well on others. Therefore, population-based testing and assessment are needed to evaluate the effectiveness of a certain procedure or material. Statistics is a science that deals with data, drawing general conclusions about the broader population from a sample of the data. Statistics can provide us a less biased, a more accurate clinical perspective than personal experiences. Therefore, as clinicians, we need to understand at least the basics of biostatistics. After completing this ITI Academy module, you shall be able to describe the source and effects of natural variation in biological populations, describe measures of variability and central tendency, define a null hypothesis, and describe the means of testing statistically significant differences, and recognize potential problems in the interpretation of study results in published literature. Variation is mostly the result of sexual reproduction. In this process, male and female reproductive cells are produced by meiosis, and these cells have half the normal number of chromosomes. In humans, these haploid sperm and egg cells have 23 chromosomes each. When these reproductive cells fuse during fertilization, they form a diploid embryo that shares the genetic characteristics of both its mother and its father. Thus, normally, no two individuals will be genetically identical. This phenomenon can occur in monozygotic or identical twins who develop from a single embryo that splits shortly after fertilization. But this is uncommon, and moreover, even in these individuals, other factors may contribute to cause some variation. Modern research is showing that junk DNA, that is, the genetic material that exists between genes, can influence the expression of genes. This epigenetic activity can also contribute to variation. The other means by which natural variation arises is through mutation, when external factors such as radiation exposure cause changes in parts of the genetic code. Such mutation processes are the main way that asexually reproducing organisms develop variation. Differences between individuals affect how they interact with their environment, especially environmental changes that threaten the survival and reproduction of groups of individuals. Some members of the population will be better able to survive and reproduce, so that their descendants may be, on average, better suited to the changed environment. Two 19th century British natural scientists, Charles Darwin and Alfred Russel Wallace, independently recognized this effect of variation and how it could lead to changes in species. They jointly released an outline of their observations in 1858. Charles Darwin, who had identified this process some years earlier, but had not published his results for fear of religious opposition to his theory, became the more recognizable founder of this new science of evolution when he published his major work on the origin of species a year later. The natural variation they described also affects the way that our patients respond to the diseases that they contract and to the medications and other therapies that we provide to them. Natural variation key learning points. Natural variation is the outcome of sexual reproduction, epigenetic activity, and mutation. Response to disease and treatment will vary among patients, in part due to natural variation. In biostatistics, a variable is a characteristic or property that can have different values in different persons or objects. For example, blood pressure and crestal bone height are variables that differ among patients. In research, a population is the set of all the values that a variable may have. This is sometimes termed the population of interest. For example, in a study comparing crestal bone loss around one-piece and two-piece implants, the population of interest would be patients receiving such implants. A sample is a subset of a population. Researchers use the data or observations from the sample to make inferences about the population of interest. For example, researchers estimate the sample mean to tell them about the true population mean. The sample mean may not be the exact population mean, but it might be very close. How close? This depends in part on sample size. In general, for a given characteristic, larger samples provide a closer representation or estimate of the population. Sample size will be discussed further in the following slides. Researchers gather data to measure variation. The nature of this data determines which statistical tool should be used to analyze the data. The three general types of data are categorical, ordinal, and continuous. Categorical data falls into different categories such as gender, ethnicity, eye color, or hair color. No category is better or worse than the other. For example, this graph shows the ethnicity distribution of students at a university in the United States. Binary or dichotomous data is an important subset of categorical data. Only two possibilities exist, such as disease or no disease. Outcomes such as caries versus no caries, or implant survival versus failure, are commonly reported in dental literature. Ordinal or rank data consists of categories, but unlike categorical data, with ordinal data, one category is better than another. In dentistry, some disease measures use numerical values or scores to represent or code for a standardized description of an observation. The Basic Periodontal Examination, or BPE, uses the numbers 0, 1, 2, 3, and 4 to represent various degrees of disease presentation. A similar system is used in the Community Index of Periodontal Treatment Needs, or CIPTN. It is important to note that although these numbers represent a definition, they have no arithmetic function. Arithmetic calculations, such as defining a mean value, have no validity. Moreover, we cannot say that a BPE score of 4 is twice as bad as a score of 2. An extension of ordinal data is quantitative data, that is, precise numbers that have arithmetic meaning. Quantitative data can be discrete or continuous. Discrete data can only be whole numbers, such as the number of cancer patients treated by a hospital each year. Continuous data, on the other hand, can include virtually any measurement. Data can be meaningfully subdivided into smaller and smaller increments and still remain valid. Examples of continuous datasets include height, weight, periodontal probing depth, and change in crestal bone height. When a given set of observations is based on continuous data such as height or weight, the data can be summarized in order to define the population with respect to that parameter. Measures of central tendency are summary statistics that describe a dataset using a single value that represents the middle or center of the distribution of values. Consider a dataset of 7 periodontal probing depth measurements that range from 3 mm to 6 mm. The mean value, or arithmetic average, is determined by adding all of the individual values and dividing that sum by the number of observations. The main disadvantage of using the mean to describe a population is that it's particularly susceptible to the influence of outlying measurements, that is, measurements that are much greater or much smaller than the next nearest observation. The median is the 50th percentile, or the middle value, when all observations are ordered from least value to greatest. Half of all observations will be below the median, and the remainder above the median value. Finally, the mode is the most frequently occurring observation. The variation in a dataset, that is, the spread of observed values, is called the standard deviation. More specifically, the standard deviation is a measure of the breadth of the data in relation to the mean. The 3 samples in this graph all have the same mean, but differ in respect to their standard deviations. The degree of spread in the dataset will have an important impact on our ability to determine if 2 populations differ. Most statistical tools used for data analysis are based on the expectation that the data is normally distributed. For example, height, blood pressure, and IQ scores all follow a normal distribution. A graph of normally distributed data forms a bell-shaped curve defined by its standard deviation, or degree of spread, designated by the Greek letter sigma, and its mean, designated by the Greek letter mu. By definition, a normally distributed sample will be symmetrical about its mean, and the mean, median, and mode have the same value. Any skewing of the data will indicate that the sample is not normally distributed. In this graph of a skewed distribution, we can see that the measures of central tendency differ. In such situations, the median is considered to be a better way of describing the average observation. It's important to note that normal distributions are based on quantitative, continuous data. Looking once more at the graph of the ethnicity distribution of students at a United States university, you can see that this data is descriptive only and does not fit within a normal distribution. Additionally, measures of central tendency have limited validity for categorical datasets. Calculation of the arithmetic mean is only valid for normally distributed quantitative datasets. In the ethnicity distribution chart, we cannot meaningfully compute an average ethnicity because the resulting value would lie outside of the categories listed. The modal value, in this case students of Asian ethnicity, would be the best descriptor of central tendency, but even this would still not be an ideal description of this population. A normal distribution provides a probability distribution about the mean value. The standard deviation is used to determine the proportion of values that fall within a given number of standard deviations from the mean. Approximately 68% of values will fall within one standard deviation of the mean. 95% of values will exist within 2 standard deviations, and 99.7% of observations are within 3 standard deviations of the mean. Thus, as we get further from the mean value, an observation becomes less likely, especially beyond 2 standard deviations outside the mean. Measuring variation and central tendency, key learning points. Results from the study sample are used to make inferences about the population of interest. Data can be categorical, ordinal, or quantitative. A set of continuous quantitative data is summarized by measures of central tendency, the mean, median, and mode. The standard deviation measures the spread of data in relation to the mean. Data is most commonly scattered in a symmetrical pattern, known as the normal distribution. In a normal distribution, 95% of the observations are within 2 standard deviations of the mean. In an experiment or study, there is an effect or difference between groups that the investigators are testing, such as the effectiveness of a new drug or the amount of crestal bone loss around a new implant design. Of course, there's always the possibility that there is no difference or effect. This lack of difference is stated as the null hypothesis. It states the exact opposite of what an investigator or an experimenter predicts or expects to find in their study. Essentially, the null hypothesis is the proposition that there is no association or difference between variables or subpopulations. The alternative hypothesis states that a relationship or difference between the factors does exist. It's easier to disprove something rather than to prove something, so most research studies are designed to disprove or reject the null hypothesis. Let's say that we're interested in implant survival following a type 2 early implant placement protocol. Our null hypothesis would then state that the timing of implant placement has no effect on the final treatment outcome. If we express this as a Venn diagram, we can see that the control group, that is, the group of patients receiving implant treatments, contains all members of the test group who may have been treated using a type 2 placement protocol. The alternative to the null hypothesis is that the two groups are distinct and different. That is, the timing of implant placement does influence the final outcome, and this can be expressed in the same manner, showing that the test group differs from the control population. When an experiment or research study is conducted, the results may show that the outcome in the test group differs from the outcome in the control group. However, it's always possible that there is no difference between the groups. That is, the null hypothesis is correct, and the patients in the study responded in the way that they did for some other unknown reasons, or because of random sampling error. Therefore, a statistical test is used to produce a p-value, which is the probability that the sample data are consistent with the null hypothesis. In other words, the p-value measures the likelihood of the collected data or observations given that the test group is the same as the control group in the experiment. A p-value is a mathematical expression of the strength of the evidence against the null hypothesis. The smaller the p-value, the stronger the evidence against the null hypothesis. A low p-value means that the data are unlikely if the null is true. Generally speaking, the difference between two groups, such as the test and control groups, is only considered to be significant when the p-value is less than 5% or 0.05. While somewhat arbitrary, this 5% level indicates that the observed outcome lies more than 2 standard deviations away from the mean. Lower p-values indicate higher levels of significance. Lower p-values indicate that the observation is even further removed from the population mean. Another concept to consider is whether a given study or investigation has adequate statistical power. This is the ability of the investigation to accurately detect a statistically significant difference between two groups or population samples, if in fact there is a difference to detect. That is, statistical power is the probability that the investigation will correctly reject the null hypothesis, given that the alternative hypothesis is true. While a number of factors influence power, the following 3 are the most significant. First, the expected size of the measured difference between the test and control groups, known as the effect size. Here, larger differences tend to lead to higher statistical power. The accuracy with which the effect can be measured, or more importantly, the level of error or spread in the data. These issues might be represented by the standard deviations of the groups, or by confidence intervals as discussed on the next slide. Smaller errors lead to more powerful results. And finally, the sample size of the groups being tested. Here, larger samples lead to greater power. Thus, as a general rule, if you're trying to detect small differences between groups, the study needs larger samples to be able to measure the effects being studied with greater accuracy. While superficially similar and often confused, the standard deviation, standard error, and 95% confidence interval all measure different aspects of a population. They are not interchangeable. As we've already seen, the standard deviation is a measure of the degree of variability in a sample, that is, the dispersion of the data about the mean. The standard error is a measure of the degree of error involved in calculating the mean of a sample, That is, the amount of variation we can expect in measuring the mean for a given sample size. The standard error is calculated by dividing the standard deviation by the square root of the sample size. Finally, a confidence interval is an estimate of the likelihood that the true mean of the population lies within the defined interval. Thus, with a 95% confidence interval, we can be 95% certain that the true mean of the population will fall into the range of values defined by the interval. Like statistical power, it's influenced by the sample size and the precision of measurement. A narrow confidence interval indicates a greater level of precision in estimating the mean. Larger sample sizes will also lead to narrower confidence intervals. There are many types of statistical tests available. In fact, too many for us to adequately discuss in this learning module. Suffice to say that each type of test will have its strengths and weaknesses that will limit its applicability to specific situations. Thus, the type of test chosen to analyze the data from an investigation will depend on the type of data that is collected and the outcome that is being tested. In a broad sense, there are three general domains for statistical analysis in dental research. Tests that evaluate differences between groups, either a test group and the broader population, or between two groups that are subject to different conditions. Tests that look for patterns in the data that imply associations between groups. Correlation tests look to see if the variables being measured vary dependently. That is, if a change in one variable will be reflected in a predictable way in another measurement. Regression tests examine whether a particular mathematical model can be used to describe the data. Tests that analyze time to event or survival data, such as time to failure of an implant or prosthesis. Comparing populations, key learning points. The null hypothesis states that there is no difference between two groups, such as the test and control groups. Investigators seek to disprove the null hypothesis. A p-value is calculated to express the strength of the evidence against the null hypothesis. A study needs adequate statistical power, that is, a large enough sample size to detect a difference between groups if a difference exists. For a given set of data, a 95% confidence interval is calculated to provide the range of values where we are 95% confident the true population mean lies. The design of an investigation can influence the potential for error when using statistical results to guide clinical decisions. In considering this potential for error, some research study designs are considered to be less prone to bias than others. Bias is anything that introduces a difference or trend that distorts, or has the potential to distort, the results or interpretation of a study. The different research study designs can be arranged in a so-called hierarchy of evidence or pyramid, in which higher levels of investigation are more reliable because their design ensures a lower risk of bias. Some common sources of bias in study design include selection bias. Here, the way that study subjects are selected may have an unexpected influence on the outcome. An example of this might be a study on oral hygiene effectiveness that employs dental students as its test subjects. These subjects might be more adept at their home care or more motivated than the general populace. Consequently, the results of this investigation might not be applicable to the population in general. More subtly, confirmation bias relates to the way that an investigator might subconsciously design their tests or interpret their results to confirm their expectations or beliefs. Emphasis on data that confirms expectations and discounting of data that falls outside of what is expected are both examples of confirmation bias. Attrition is another hidden source of bias. Few clinical studies that continue over a number of years will keep in contact with all of their subjects. Some will die, some will move away, and some will just decide that they no longer wish to take part. These dropouts can affect the quality of data, especially if the dropouts have a disproportionate representation of a particular trait. For example, if the dropouts are mostly subjects that have had a treatment failure, this outcome might be underreported in the study. In other words, systematic differences between people who leave the study and those who continue can introduce bias into the study's results. Confounding bias occurs when a third variable, not controlled for or considered by the study, has an effect on the outcome. Many years ago, studies identified that coffee drinking was correlated with an increased risk of coronary artery disease. These studies did not ask their subjects if they also smoked cigarettes, nor did they control for a smoking habit. Thus, those coffee drinkers who were also smokers had an uncontrolled effect on the data and on the conclusions that were made from this data. Finally, publication bias is common. Researchers are more likely to publish papers that find strong effects for a given treatment, but are less inclined to publish papers on experiments that find equivocal or negative outcomes. Editors of journals may also be less inclined to publish papers with negative or equivocal findings. Additionally, sponsors of research may be tempted to resist publication of results that are negative or neutral. In a review of the results of investigations into the effectiveness of selective serotonin reuptake inhibitors in children with depressive illness, Guzner and others found that research projects sponsored by drug manufacturers were much less likely to report negative or neutral findings than non-industry sponsored projects, and that both of these were less likely again to report negative or neutral outcomes than unpublished data reported to the US Food and Drug Administration. So, how do we decide if the results of a study and any conclusions being drawn from those results are valid? In other words, how likely may a study reflect true outcomes? A simple checklist that looks at some of the information discussed in this learning module might help. What type of study are we looking at, and where does it lie in the hierarchy of evidence? Is there a control group? Is the study prospective, so that it looks for issues as they arise? Or does it only report retrospectively on what was seen in that group? For clinical studies, how long was the follow-up, and how were dropouts managed? That is, were they accounted for in the statistical analysis? How big was the study sample, and how are these subjects selected? What statistical tests were used, and were they appropriate for the type of data and the type of information being sought? And finally, were the conclusions drawn by the investigators supported by their results? By understanding the statistical process involved in research and applying these criteria, we can be a little more critical in our search for information that will guide us in how we treat. Potential for problems, key learning points. The different research study designs are arranged in a hierarchy of evidence to reflect their potential for bias. When applying study results to clinical practice, it's important to consider the effects of bias and confounding. A quality checklist is useful for evaluating the validity of study results and conclusions. An Introduction to Biostatistics, Module Summary. Patients respond to implant treatment differently because of natural variation in the population. Investigators gather data on samples of the population and then make inferences about the broader population using the sample mean and standard deviation. For a given set of sample data, a small p-value allows investigators to reject the null hypothesis. For example, the hypothesis that states that a treatment has no effect. A narrow confidence interval indicates a more precise estimate of the population sample. When applying the results of a research study to clinical practice, it's important to consider the quality of the study and its potential for bias.